Last but not least, Alan Goodman, who's going to tell us about host-produced Eiger, TNF, and the bacterial type 4 secretion system that enables susceptibility of the fly to Coxiella burnetii infection.
So sorry for that long title. I went to a little workshop by Mark Johnson yesterday, and I think he'd be pretty upset with this title. He's the editor-in-chief of Genetics. So I'm going to tell you about our development of this model system in flies for Coxiella burnetii. A little bit of background about Coxiella burnetii. It's a gram-negative obligate intracellular bacterium, and it causes the zoonosis called Q fever, which stands for query fever, because at the time of its diagnosis in 1937, they didn't know what was causing this disease. Infection occurs at a very low dose, and there's high morbidity. Back in the 40s, there was a thing called Operation White Coat, where they were infecting military personnel to figure out what is the lowest possible dose to cause this disease, and it's as little as 1 bacteria. So because of this high morbidity and infection at low dose, among other things, it's a Category B bioterrorism agent and a select agent, and it necessitates BSL-3 conditions for work. The target cells for Coxiella are monocytes and macrophages, among others. So here's a picture of a macrophage freeze fracture. You can see Coxiella inside this macrophage. So there's 2 forms of Coxiella actually. There's the phase 1, which is this pathogenic form, and contains this long O antigen, and this is the BSL-3 pathogen. So there's also the phase 2 form, which does not contain this long O antigen, which is one of the main virulence factors of Coxiella. So in 1986, it was cloned this what was called 9-mile phase 2 strain, which can be used in BSL-2 settings. So some models for Q fever and Coxiella. Of course, there's animal models, and this is again with the BSL-3 agent, guinea pigs, mice, non-human primates, and its natural host reservoirs in goats, sheep, and cattle. So phase 1, as I mentioned, is a BSL-3, needs BSL-3 labs and animal facilities. So there's a need for these BSL-2 models. There's one model to use to study 9-mile clone 4, and that's Galleria melonella, which is the wax moth larvae. But these in vivo models in BSL-2 can be used to study sex-related susceptibility, the low infectious dose needed, and chronicity. So we want to propose and ask if we can use the Drosophila as a model for Coxiella infection, and specifically, can we use Drosophila and 9-mile clone 4 in BSL-2 conditions to study Coxiella? So to begin, we asked if 9-mile clone 4 can infect Drosophila hemocyte-derived S2 cells. So here we show a graph of S2 cells in red and sheep macrophages in black. So you can see that Coxiella 9-mile 2 grows a little slower in S2 cells than it does in sheep macrophages. We look for the presence of the Coxiella-containing vacuole. So here you can see this large vacuole, and here's the nucleus. So that's a very representative of Coxiella infection. Then we did a gentamicin protection assay. So in the absence of gentamicin, you see very rapid growth. So this is using a GFP-labeled strain. And you see very fast growth from 2 to 10 days in the absence of gentamicin. But when gentamicin is added very quickly after infection, you see very little growth. But when it's added a day after infection, because it's protected in the vacuole, you can still see nice growth of Coxiella 9-mile phase 2. So then looking just again at some antimicrobial peptide inductions, because it's a gram-negative bacteria, you can see induction of drosocin very rapidly by 24 hours post-infection, and less induction, not significant induction of defensin, typical toll readout. So next question is, are adult Drosophila susceptible to 9-mile clone 4? And remember, it's not, this phase 2 strain is not virulent to immunocompetent mammalian hosts. So looking at 2 different doses, 100 or 100,000 bacteria, you can see that it causes mortality in both females and males. And heat-killed bacteria, in green here, does not cause significant mortality. Next question is, does it replicate in adult flies? So again, in both males and females, by 30 days post-infection, you can see a high increase in titer. And I want to point out that we're measuring here what's called genome equivalent. Coxiella is very hard to measure colony forming units. So what we do is by qPCR measure genome equivalent of the .A structural protein. And also in adult flies, we do see induction again of drososin, but not significant, if any, induction of defensin. So again, an IMD pathway readout as opposed to the toll pathway. So before we started to look into mechanisms of mortality, we wanted to ask, what kind of cells are hemocytes specifically being infected with this 9-mile clone 4? So the experimental design was to infect third instar larvae with the GFP reporter on hemocytes in a pool of high titer 9-mile clone 4 M-cherry labeled using a tungsten needle, and then 20 hours post-infection, we'd extract hemocytes onto cover slips, fix them, and stain with DAPI. So here we're looking just first at a Z-stack, and you can see the presence of bacteria by the white arrows. And now we're going to look at just first a surface rendition of the bacteria, of the cell with the bacteria inside. And then we'll take cross-sections along to show the presence of the bacteria inside the cell. And I want to point out that these bacteria are normally a 0.5 micron to 1.5 micron in length. So you can see here there's one extracellular bacteria. So looking at the cross-section, again, we can see indeed intracellular bacteria, and this is very soon after 20 hours post-infection, so we do see the presence of bacteria inside these cells. And again, just moving through the cross-section, we see 3 separate instances of bacterial infection in these primary hemocytes. So again, now looking at the mechanism underlying the susceptibility of adult Drosophila to 9-mile clone 4. So because we saw induction of the IMD pathway, we wanted to ask if IMD mutants might be more susceptible, and indeed they were. So looking at relish mutants or PGRP, LC mutants, we see increased mortality as compared to W1118 flies. However, we didn't see a difference in bacterial load among the 3 different fly strains. And we did see decreased induction in the 2 mutant strains, especially in the absence of functional relish, no induction of drososin, and again, high induction in the wild type or the W1118 strain. So take-home message is that mutations in the IMD pathway render flies more susceptible to 9-mile phase 2, 9-mile clone 4 infection. So previously, it's been shown that Eiger, the only TNF homolog in flies, can have different effects on bacterial infections depending on the type of bacteria. So here we're looking at Eiger mutants, and we show that the absence of Eiger, really a mutation in Eiger, causes better survival of the flies as compared to W1118. And then we did an RNAi experiment, so knockdown of Eiger. So knockdown of Eiger is, again, also protective as compared to the control flies. But again, we didn't see a difference in bacterial replication in these Eiger mutant flies as compared to the control flies. So again, take-home message here is that the loss of Eiger protects flies from 9-mile 2 in induced mortality. And this is, again, similar to Salmonella and mycobacteria infections. So during those infections, loss of Eiger is protective. And also a recent paper showed that Coxiella is able to inhibit a host inflammatory response, IL-1 beta, and a TNF alpha response in bovine macrophages. So that's looking at the host side. Now we want to look at the bacterial side of things and ask if there are other virulence factors of the bacteria that may cause susceptibility of the bacteria, susceptibility of the host to infection. So another major virulence factor of Coxiella is called the type 4 secretion system. And this is a complex of proteins that is there for the secretion of effector proteins into the host cytosol. So there are structural proteins here, and there are also regulatory proteins of these structural proteins. So PMRA is able to regulate a number of these structural proteins. And we're going to look at 2 different mutants of Coxiella clone 4, 9-mile clone 4, specifically those lacking functional .A and those lacking functional PMRA. So here looking at, this is NS2 cells, so we see the background strain can grow to high titer, and the other strains do not grow to as high of a titer as compared to that background strain. And here looking at a Western blot using a polyclonal antibody. So we have, again, a really large non-specific band in the S2 cells. But we see, so here, this is the background strain. And then these mutant strains have so lower expression of the Coxiella proteins. And then by 12 days of post-infection, you can see the absence of these in the 2 mutant strains as compared to the background strain. So then using GFP-labeled versions of these strains, again, with the background strain, we see a very large Coxiella-containing vacuole, a single vacuole, and then these in 2 mutant strains, we don't see very good formation of a large vacuole, and again, disrupted vacuole formation. And then looking in adult flies, these are Oregon R flies, we see that, again, the background strain can kill flies very rapidly, but these 2 mutant strains do not cause significant mortality in the flies. So the type 4 secretion system is required for replication and pathogenesis in Drosophila. And again, this is similar to the role of this type 4 secretion system in mammalian systems as well. So just to summarize, Coxiella clone 4 infects, replicates, and induces an immune response in S2 cells. The adult flies are susceptible to this attenuated strain. Fly mortality requires the presence of live bacteria, and it elicits antimicrobial peptide response from the IMD pathway. The loss of the IMD pathway renders flies more susceptible, while the loss of Eiger is protective, and the type 4 secretion system is required for pathogenesis. So I'll take some questions now, and thank you for your time.
Two very quick, very basic questions. The first one was, when you saw the bacteria in the cell, in the hemocytes, you say, is it infecting the hemocyte, or is it actually the hemocyte is phagocytosing it?
So it's taken up the bacteria. We haven't been able to have our cells survive long enough, these primary cells, so that we can see replication of the bacteria in the cells. Just a technical issue that we're having trouble with.
Okay, and the second question, when you delete either the IMD pathway or the Eiger gene, are you tissue specifically knocking it down, or are you doing it globally?
Yeah, that's using for the Eiger stuff, it's an actin Gal4 driver.
Okay. Do you think there is going to be a difference if you do tissue specific, and would you get a better protective or no protective?
That's a great question. We haven't tried other drivers. I think using the hemocyte specific driver would be a very good experiment.
Thank you.
Do you know which receptor might be involved in the recognition and engulfment of those bacteria?
The recognition of
Coxiella?
So it's likely taken in by phagocytosis, yeah. But of how it's binding to the cell, we don't know.
Any more questions? Hi, just a quick question. So I know that there are other bacteria which have type 3 or type 4 secretion systems can induce a starvation response in their hosts by depleting, extracellular nutrients and so on and so forth. Do you think that the activation of the Eiger pathway has something to do with a similar starvation response in your strain, or is it a different response, if you have a sense of?
Yeah, so I mean, as you mentioned, it's the formation of this vacuole and the activation of that secretion system. That's what causes such high morbidity in humans. So if it's depleting nutrients in the fly or the cells, we don't know, but it's a good experiment.
All right. I'd like to thank all the speakers for a terrific session. I'd like to thank also my co-chair, Jean-Marc Reichard from the University of Strasbourg, who's been sitting there very quietly, and especially Dalima for helping me chase the speakers around for the upload of their presentation. And one final quick announcement. We've had very, very few abstracts this year for this particular session. I strongly encourage you to send abstracts for oral presentation. There was only 14 abstracts to select 8 talks. So your chances of getting a talk is very high. So please try to send us abstracts for oral and poster presentation. We really want to see that session continued at this meeting. Thank you very much.